Hi everyone. Today we are honored to have Dr. Ben Cowling from the University of Hong Kong to talk about repeat vaccination effect for influenza. We will have 10 minutes Q&A after his talk. If you have any questions, please type in your question to the Zoom chat or ask it in person after the talk. Without further ado, please take it away, Dr. Cowling.
Thank you very much. I'm Ben Cowling. I'm a professor of infectious disease epidemiology at the University of Hong Kong, where I've been for 20 years. I went there in 2004. The Hong Kong government had this idea that after SARS-1 in 2003, they should get ready for SARS-2 at some point in the future. And then it did eventually come 20 years later almost. And the team that we had in Hong Kong did quite a lot of early interesting work on COVID. But my talk today is about flu. It's about repeat vaccination effects, which is something I've been interested in for quite some time, more than 10 years, since my first paper on repeat vaccination effects. And it's an interesting phenomenon. So going back even further, there's a very interesting paper by Hoskins and colleagues published in The Lancet in 1979, reporting an observation that children who got vaccinated in a particular year and also had previously been vaccinated seemed to have a higher risk of infection than those who were only vaccinated this year for the first time. So the repeat vaccinees didn't do as well as the first-time vaccinees. And there were some hypotheses about why that might be happening. And it worried the US government enough that they commissioned a large trial, a 5-year trial reported by Wendy Kaito and colleagues in 1997, some years, of course, later, which in the first paper, I suspect if you look at it carefully, that the findings are not quite supported by the data presented. So they talk about repeat vaccination effects, but it's not quite clear to the strength of the effect. And then in the Kaito paper, they say there's nothing to worry about. There's no real repeat vaccination effects. But actually, if you look at the data, there is. So it's kind of fascinating to think about what's happened since then. When we talk about repeat vaccination effects, we're talking about clinical protection, that repeat vaccinees some years sometimes seem to be less well protected against the disease influenza, but there's also quite a number of studies of immune responses to repeat vaccination because that's part of the presumed mechanism. And the studies I'm going to present are studies of immune responses rather than of clinical protection because of the size required. A concrete example of repeat vaccination effect in more recent years from a Canadian group, Danuta Skowronski and colleagues, finding that in the year 2014 to 15, people who'd only been vaccinated for that season but not in the preceding year had quite good protection against H3N2. But those who were vaccinated in both years, so the current year and also the preceding year, were possibly at higher risk of H3N2 than those who not been vaccinated either year, which is kind of concerning because we would hope that every time you get flu vaccine, you're protected for that year. And this particular study suggested that in some years, people who get vaccinated may not, repeatedly at least, may not be protected for one particular season. An important thing to remember, though, with this kind of data is that the prior year vaccination was protecting people in the prior year. So you have to think about a longer time scale. And one of the things I've been thinking about in my research is not necessarily just 1 year at a time, but about vaccine programs and how much disease is prevented by programs in the longer term or in the medium term, rather than just on a single year basis. What are the reasons why these repeat vaccination effects occur? A number of hypotheses in the literature, and I'm not an immunologist, so not the world expert on the immunological mechanisms, but the first hypothesis written here is the idea that if you've already got a lot of antibodies in your system, they might be able to bind to the vaccine antigen before your body even gets a chance to respond to the vaccine antigen. So it's like blocking the vaccine before you have a chance to respond. And there's something similar that's thought to happen with the live attenuated influenza vaccine, the nasal spray vaccine, that if you've already got a good level of mucosal immunity against flu, when you get the nasal spray vaccine, it just doesn't cause an infection. So you don't get much of a response to it in that case. So that's a situation where if you've got a lot of immunity already, the vaccine might not work as well, perhaps. The second hypothesis is related to antigenic seniority or imprinting in some sense, in some kind, where if you get repeatedly vaccinated, you get a good response to the older strains, maybe not such a good response to the very latest strains. I'll come to that at the end of my talk. And then a third hypothesis is infection block, where if you got vaccinated last year, maybe you didn't get flu last year because you were vaccinated, but if you got flu last year, maybe you wouldn't get it this year. And so there's infections in the mix that are messing things up. And one of the exciting things in the second study I'm going to present today is that we don't have any infections in the mix. And so we can look purely at repeat vaccination effects without having to worry about infection block. And then there's some other things that might be going on that we don't quite know. So I'm going to present data from 2 studies I've been running in Hong Kong. I've been doing a number of flu vaccination trials. And so these are 2 of them. There's others that I won't have time to talk about. The motivation for my study of twice annual vaccination was the observation that in Hong Kong, we have flu for much of the year. And this is pre-COVID pandemic, where we tended not to have flu seasons between September and December, but any other time of the year was a possibility of having a flu season. And so whereas in the US or Europe or other places in temperate locations where the flu epidemics tend to only occur in the winter, we can get epidemics at other times of the year. And so a vaccine which provides a shorter duration of a good level of protection might not be as ideal as a strategy or a vaccine which provides longer protection. So I'm not saying that the flu vaccine only lasts for 6 months, but there's some evidence that waning occurs over that type of time scale. In Hong Kong in 2015, there was a big H3 winter season, and then reports from other parts of the world that there was a new strain of H3N2 circulating, which was in the updated Southern Hemisphere vaccine, but not covered by the Northern Hemisphere vaccine. And so after that first wave of H3N2, the government decided to do a spring booster campaign with the Southern Hemisphere vaccine, which had the updated strain. So it's not the wrong vaccine, it's the mismatched strain that circulated last winter. And they vaccinated quite a lot of people. And then the new H3 strain did actually circulate. It was one of the situations when what you worry about actually does happen. And so the Southern Hemisphere campaign probably did do quite a bit of good. We looked at an observational evaluation of that, but we were already at that time planning to do a trial of twice annual vaccination as a way to solve this protection for year-round protection. Sorry. And then I was very interested a few years ago to read an article in Science by John Cohen, reporting on this character, Stanley Plotkin, who got a late season booster. You can read it on the slide. And I know Stanley Plotkin, so I was surprised to see his name. How come this guy's talking about getting a late season booster? Because he's like the expert vaccinologist in the US. He's written the textbook on vaccines in the US. So if he's getting a late season booster, you can see it in the snippet of text. So 3 months after his first flu shot, he got a second flu shot, so twice a year. And he thought it made a lot of sense to give him this late season booster and protect against maybe a late season flu infection. So I thought it's not only places like Hong Kong that are thinking about year-round protection, it may be relevant in other parts of the world. So we created a randomized trial in older adults, and the diagram is here. The syringes of vaccination from either the Northern Hemisphere NH or the Southern Hemisphere SH formulation, and the red group get 2 doses per year, once in the autumn, once in the spring, and the once annual group in blue get the winter vaccine like everybody else in Hong Kong in that age group, but they don't get the Southern Hemisphere, they get a placebo instead. And so the blue is like the routine practice for older adults, and the red is double the doses, essentially twice a year doses of flu vaccination. The study was designed with 400 people per group, and of course there's dropout for various reasons through the study. So we did add some more people in year 3 to the trial, but it's no need to get into too much detail about that. The vaccine strains were updated from time to time, as happens. So we have H1, H3, B Victoria, and B Yamagata. B Yamagata didn't change throughout, but the other strains were updated. And altogether, I think it's 15 strains in the 5 years. So it's typically in a given year, there was at least one strain update. And it's interesting, sometimes the update came in the Southern Hemisphere vaccine, sometimes in the Northern Hemisphere vaccine first. So what happened is the primary results, these are the antibody titers against the vaccine strains, which were changing over time. So that's why there's discontinuities between these lines. The antibody titers against vaccine strains, a higher level is better, a lower level is not as good. And the open circles in blue are placebo, where we expect like a horizontal line because it's a placebo. And then the others are the day 0, day 30 pairs, where we expect a boost from vaccination. And I don't want to get into the details of every single strain in every single year. I just want to give you a big picture interpretation. Red is above blue, basically. Like there's very few places where red is below blue, very few. And then we can look at it in different ways with different outcome metrics. This is just the GMT, but there's other things, the proportion above 40, and whatnot. But the big picture interpretation is that red is typically above blue, or maybe the same in some of the periods. And so my conclusion from the antibody data is that twice annual vaccination would either give a similar level of protection or better protection than once annual vaccination. And I'm very glad that this is what we found, because when we designed the study at the beginning of the trial, we were concerned that there'd be a type of over-vaccination effect, where if you give people too many vaccines, their response attenuates more and more and more. And we were worried that red might be above blue at the beginning, but by the end, red would be worse because there'd be such an attenuated response. And so we'd be almost harming people in a sense of over-vaccinated. So that doesn't seem to be the case. People seem to be doing just fine with the twice annual. The next question is, what about the clinical benefit, the clinical differences? This was only 200 people per group. We don't have a large amount of PCR-confirmed infections. We have some, and we have very few flu hospitalizations. But given what we understand about the relations between antibody titers and clinical protection, my inference from some rough look at these levels of antibodies and rough averaging over the years is that maybe a 10% or so difference in clinical protection. So the red group might have 10% fewer flu cases than the blue group. And that's not a massive effect. And that's just a very rough inference. We'll do a better job, a proper job in due course. So maybe it'll come out more or less than 10%. But if it was 10% for residents of long-term care facilities in Hong Kong, I mean, that could correspond to quite a cost-effective solution for providing better protection against flu. And for Stanley Plotkin, would seem to be quite a good idea. It's not an expensive vaccine to get. Now, I want to talk a little bit more about repeat vaccination effects. So one of the phenomena observed in flu vaccination studies is that if you have a higher titer when you get vaccinated, if you have more antibodies when you get vaccinated, you don't respond as well to vaccination. So the curve, the red line shown on this plot is called ceiling effect. It doesn't mean it's hit the ceiling. It's just called a ceiling effect for some reason. So the higher level of antibody you've got before vaccination, the weaker your response to vaccination. And we see this kind of a bit flattened response compared to the diagonal line. And this has been observed many, many times in vaccine studies. Some people think the repeat vaccination effects are just ceiling effects. And that was, I think, the first hypothesis on the slide I showed at the beginning. But actually, repeat vaccination effects are thought to be something slightly more than that, where even for the same level of baseline titer, we see an attenuated response to vaccination. So if someone who comes in with a titer of 40 and gets vaccinated, their response will be stronger if they're a first-time vaccinee compared to if they're a repeat vaccinee, for that baseline titer. And explaining the difference between red and yellow on this figure is like what I want to do next, but it's challenging. So why do people have attenuated responses? And the data from RETAIN show repeat vaccination effects. So we've plotted from beginning to end the 5 years of the data that I've shown you, the earlier years are in lighter shades, the later years are in darker shades, and the slope is flattening. Basically, for H1, for H3, and for the two B strains, the slope is flattening. You see it most clearly on the right-hand side for B Yamagata, where the vaccine strain has not changed throughout the 5 years. It's been the same vaccine strain throughout. And by the end, the lines are very flat and very close to the diagonal. At the same time, we have more and more observations at high levels of titer. So in a way that people are already quite well protected, most likely. Not so critical that they don't have a good vaccine response, but it's certainly interesting, and there's a lot of room for further study. So this is the first study. Conclusion from the observations we've made so far is that twice annual vaccination probably will induce certainly higher antibody titers, most likely additional protection. There's no evidence of a reduced protection during the first half of the following year. It wasn't that red was above blue half the time and below blue the other half. It was basically the same or above blue, red. So the implication is that I think there's certainly good justification to do more studies of twice annual vaccination. There's only one other study of twice annual vaccination to my knowledge in Singapore, reported in CID a few years ago, with either 2 or 3 rounds of twice annual vaccination, plus my own observation study from 2015 published in CID. So that not a trial. So I think there's grounds to do maybe a larger study in long-term care to see whether twice annual is a good solution. Obviously, it doubles the cost compared to vaccinating once a year, but flu can be nasty in long-term care facilities. And so I think there's a good rationale to pursue this as a vaccination strategy. And for the US, maybe late season boosters in long-term care facilities or vulnerable individuals might also be considered. And the other exciting news about the RETAIN study is that we were able to get funding to continue it for another 4 years. So we're going to have 9 years of data at least, and I would like to keep it going a little bit longer. So in a few months' time, we'll be giving the 18th dose of flu vaccination to the twice annual group. And we're getting all these blood samples before and after, and quite exciting to see what's happening. Now, the second study I want to talk about is a collaborative study with Sarah Covey at the University of Chicago and a number of other people I will name at the end. The name of this study is the DRIVE study, and I can remember what it stands for: Determinants of Repeated Influenza Vaccination Effects, DRIVE. So it's an exciting idea. The study diagram is shown on the right-hand side. This is a randomized trial. And at the beginning of the study, we divided the participants into 5 groups, shown in the rows, and then we planned out a schedule for them over the 5 years of the study. We are coming up to the end of the study. Later this year, we'll have year 5 vaccinations. And so you can see red is vaccination, P is placebo. At the beginning of the study, only one of the groups gets vaccinated, but every year there's one more group starts getting vaccinated. So every year from the second year onwards, we can compare the first-time vaccinees with repeat vaccinees and see what's the difference in the response for the first-time vaccinees compared to those who've been repeatedly vaccinated. We're using the Flublok vaccine rather than the standard egg-grown inactivated vaccine. Flublok is a recombinant vaccine grown in insect cells, which is a better match to circulating H3N2 strains particularly. Does not include any neuraminidase, so we can also use neuraminidase quite happily to pick up any infections that might occur. Whereas with egg-grown inactivated vaccine, there's some neuraminidase which not necessarily very well controlled in those vaccines. So there's 2 reasons why we're using Flublok. The inclusion criteria for the study: younger adults 18 to 45 years old who did not get vaccinated in the 2 years preceding the study. So they're coming fairly clean in terms of vaccination history. So we don't have to worry about repeat vaccination effects at the very beginning, and not in a priority group because those people should get vaccinated, should get placebo. And then we, as in the other immunogenicity studies, we get samples at 0, at 30, and some other time points as well. Primary outcomes are the antibody titers, like in the RETAIN study, GMTs at day 30 and day 182, and then also the boosting and other things as well. Now, one very interesting thing for the DRIVE study is that we didn't have any flu for the first 2 and a bit years of the study, first 3 years of the study. And one thing separate to my talk on repeat vaccination effects that some of you might be very excited to see is that we had no flu for 3 years. There was a very small number of detections in travelers and in recipients of the live attenuated flu vaccine who got sniffles. The next day, the kid went to the doctor, they tested, and it came back for flu, and then it turned out it was vaccine virus. So there's no flu epidemics in the community. We had very stringent COVID measures in Hong Kong from March of 2020, February of 2020, right through to early 2023. But the majority of measures were relaxed in mid to late 2022. Schools resumed normally in September 2022. Travel restrictions were gone in September 2022. Social distancing measures in the community were gone in September 2022. And it's only the mask mandate, which was kept in place for a few more months with the express rationale that it was not going to be relaxed in September, October, November, December 2022 because of the fear that there will be a winter surge and that COVID and flu and whatever else comes, we'd have a triple-demic and the hospitals would be under a lot of pressure. So some health advisors persuaded the government to hold off on relaxing the mask mandate until the 28th of February. And lo and behold, in March of 2023, flu came back. And we published a paper on this in EID on masks and flu, where we estimated that the mask mandate was reducing flu transmission by 20 to 30%, if I remember correctly. And with the RT of flu at the start of a season being in the order 1.2, 1.3, that was maybe enough to hold off flu, at least as an epidemic, until the mask mandate was relaxed. Having said that, when the mask mandate was relaxed, people didn't immediately take off their masks and never wear them again. There was still quite a bit of mask use in the community. It's maybe masks are even more effective than we imagined, because it was the effect of the mandate that we were estimating, not the effect of 100% masking versus a low level of masking. And we've had interesting dynamics with flu ever since then. So we've had H1 initially and some H3, and then we had 2 peaks of H3N2 with similar strains. And we're just right now having H1N1 again. We haven't had any B epidemics. So it's odd patterns actually for flu in the post-pandemic era. But the key point for the DRIVE study is that in the first 2 and a bit years, we didn't have any flu. And so we don't have to worry about infection block as an explanation for any repeat vaccination effects. Vaccine strains were updated in the DRIVE study for H1 and H3 in the 2nd year and for H3 and B in the 3rd year. So we're looking particularly for what happens when there's a strain update versus when the strain stays the same. In general, with repeat vaccination effects, they tend to be stronger when the strains stay the same, but that's not always the case. So I'll share with you some preliminary results just from a subset of participants that we've tested so far. We'll have more comprehensive results later this year. So I'm going to color code. The red group is the repeat vaccinees. The blue group is the placebo first and then vaccination. So here we go for H1N1 for the year 1 vaccine strain on the left and the year 2 vaccine strain on the right. You can see on the right-hand side of each of these 2 columns, each of these 2 panels, the red and the blue line cross. And that is related to repeat vaccination effects. It could also be attributable to ceiling effects. We'll have to see in a moment about the difference between those 2. But the crossing of the lines is something that we've been looking out for. So in other words, the repeat vaccination group started with higher baseline titers in year 2, but they had a much weaker response to vaccination. They ended up with slightly lower titers than the blue group. The differences in day 30 titers were not statistically significant here, but the size is a 2-fold difference. So we didn't test that many samples at the moment. Maybe we test more, and that effect persists, it will be significant. For H3N2, we did not see the same crossing of the lines, which is in itself quite interesting. And remember, the 2 years have slightly different vaccine strains. This was testing by neutralization, not by HI, because there's issues with HI testing for H3N2 strains recently. And then for the 2B strains, the vaccines were unchanged in years 1 and 2. And again, we do see the lines crossing, the blue and the blue line crossing. So this is like a proof of principle that we see something interesting in the same people that are followed over multiple years. The titers are elevated in year 2 for the repeat vaccinees at baseline, and then they have attenuated responses. So then we get to year 3, we add a new first-time vaccinee group that's going to be in green, and the blue and the red are now both repeat vaccinee groups. So for H1N1 on the right-hand side, we see the green group are the first-time vaccinees. They come from a very low level of baseline titer. They get boosted the most, but then by day 180, it all ends in about the same place. And in the placebo group, we see some day 182 titers that are higher, which is because of infections between day 30 and day 182, because flu was then circulating at that point. And there may be other infections in the vaccine groups as well. It's just in the placebo group, it sticks out because there's no other reason for them to have higher titers at that moment. And in H1N1, it's interesting that there is a statistically significant difference in this subset of samples that we tested between the day 30 titers for the green and the red group. So whereas in the year 2 data, there were no differences at day 30, in this, it does look like there is a significant difference between red and green, meaning that the people who've been vaccinated 3 times in a row potentially had reduced protection compared to the first-time vaccinees in this year of the study. So that's fascinating. For H3N2, with neutralization rather than HI assay, which could be part of the reason why we don't see the same kind of effect, the lines are much closer. And then for B Victoria, we see the green starting the lowest, but getting the biggest boost. No differences at day 30, day 180. And then lastly for B Yamagata, again, the green starting lower, getting a big boost, but then ending up in a similar place. And the titers for Yamagata seem very high. And again, on a sidetrack, you may have followed the news that B Yamagata seems to have disappeared. It's not been detected since early 2020. And vaccines for flu have been quadrivalent with H1, H3, B Victoria, B Yamagata. But B Yamagata is now presumed to have gone. And in the US and other parts of the world for the coming winter, they're returning to trivalent vaccines without B Yamagata because it's thought B Yamagata has really gone. It's not going to be back. One of the interesting things about B Yamagata is that population immunity has been very high to B Yamagata in recent years before the COVID pandemic. The antibody titers here are very high. The vaccine strain hasn't changed for years. Remember, in the RETAIN study, it didn't change for 5 years, and probably longer than that. There's a possibility that the reason B Yamagata disappeared, whereas the other strains of flu did not, is because population immunity to B Yamagata has been relatively higher. So it needed relatively less effort to eliminate. But that's a hypothesis, there's a speculation. But it is notable that the antibody levels in all my studies are very high for B Yamagata. And it's not only true in Hong Kong, true in other parts of the world as well. So another way to look at the results is the color coding has changed. This is now analyses done by Sarah Kobe and Marcos Vieira. So the color coding is slightly changed, but the columns are now the placebo group. The green is the first-time vaccinees in year 3, and the other 2 are the repeat vaccinees. And by the top is indicating the significant differences at day 30 that I pointed out to you in H1N1. So this is the antibody titers measured by the HI assay and imposing assay. And then a separate assay that we're using, a Luminex, which picks up binding antibody that's possibly like a broader or more noisy assay. The Luminex results have a slightly different picture with a lot more significant differences between the groups, which is also kind of a very interesting observation that I'm still thinking about how to explain, and maybe Sarah could have a good explanation for it. But as fast as I go back, these are the antibody titers by HI and by virus neutralization, where at day 30 above and day 180 below, the levels are pretty similar between the first-time vaccines and the repeat vaccinees. But by Luminex, there seems to be kind of more differences between the first-time vaccinees in green and the repeat vaccinees in the other 2 colors. So there's something that we want to dig into more. This is binding antibody. The other one is something that's a little bit more specific. And then coming back to this idea of ceiling effects and repeat vaccination effects, in the DRIVE study, we can create a model. The figure at the bottom is a hypothetical model. So the model we're using to explain the day 30 titer based on the day zero titer is the second and third element, with the antibody ceiling effect in the model by virtue of the regression coefficients. And then the number of vaccinations, which becomes the repeat vaccination effect as the coefficient B2, and then some other things. And so we can evaluate different models to see which ones pick up what's in the data the best, or a model without any of these effects, and see what works. And it turns out that the antibody ceiling effects and the repeat vaccination effects were both broadly supported. So these are fitted lines to the data I've shown you in the earlier slides. So this is a model fit that includes ceiling effects and repeat vaccination effects, both as significant terms in the model. And yeah, this is as far as we've got. The data are quite new. The top panel is the HI and the neutralization, and the bottom panel is the binding antibody by Luminex, where the lines seem to be more separated in some cases. So it's fascinating, I think. And I can't give you the answer to why repeat vaccination effects are occurring. I can't give you the answer today, but maybe I'll come back in the future and have an answer for you of what's really going on in terms of the mechanism. But I think this study is at least proving that there's repeat vaccination effects beyond ceiling effect, which in the literature, some people argue that repeat vaccination effects are just ceiling effects. So we show they're not. And next step is to look at the mechanisms a bit more. And one last thing I want to share with you before the end is some very interesting work by Jesse Bloom and his colleague Andrea Lowe's. There's a preprint about this analysis just recently released. Using drive samples, this is formatted in a different way to before, so I'll explain what's being shown here. We have year 2 samples from the drive study. So year 2, day 0 at the top, day 30, and then day 180. And the two groups being compared are the first-time vaccinees in year 2, so placebo, and then getting vaccinated in this year, and that's in blue. And then the repeat vaccinees are in yellow. So they got a vaccination the previous year. This is year 2 data. So they come in with a higher baseline. So yellow's got a higher baseline in year 2, and then the response is a bit weaker in the repeat vaccinees. And by day 180, it comes out to be about the same. But the things being shown in the columns are a lot of different strains. And Jesse Bloom's laboratories come up with a very clever way to do large numbers of neutralization tests in a relatively easy and efficient way. So all the things on the x-axis are different strains, including some historical H1 strains on the left, some clade 5A1 strains, which was year 1 vaccine strain, some of those in the gray shaded area on the left-hand half of the plot, and then the year 2 vaccine strains and related strains on the right-hand side, the clade 5A2 strains on the right. And so digging into the response, we can see evidence, for example, the day 30, the middle panel of this plot, the blue and the yellow lines are kind of closer on the left-hand side than on the right-hand side. So there's a repeat vaccination effect there in the sense that the repeat vaccinees in yellow are not doing as well against the new strain, but they're doing fine against the old strain. I think there's a lot more to dig out of this type of data. This is just H1N1, but I suppose we could imagine having similar kinds of data for the other vaccine strains. And this is only year 2. So we could imagine as well having years 3, year 4, year 5, and looking at vaccine strains in different years and the responses to those strains and the back boosting and the antigenic seniority and all of that. So I'm quite excited about the opportunities presented by this trial to really dig into the repeat vaccination effects. And we've been collecting whole blood as well as serum. So we have a whole load of frozen samples ready for T-cell testing as well, and other elements of the immune system, just pending laboratory testing and then analysis. So I'm nearly at the end. So for the drive study, we found repeat vaccinees had higher pre-vaccination titers. They came in at a higher level, but reduced mean-fold rises. Not many significant differences. There was difference for H1N1 I think in year 3, but generally the day 30 and the day 180 as well came out similar. So there may not be implications for protection, but there's certainly interesting immunological differences. We'll carry on looking at the samples from the study, and there's also some infection from year 3 onwards when flu returned in Hong Kong. And then one last thing, when vaccine strains are selected, there's an exercise in judging how similar different strains are from each other, often using ferrets infected with one strain, and then their blood tested against other strains to see what's the degree of cross-reaction, essentially, between antibodies that are stimulated by infection with different kinds of strains, or maybe vaccinated. You could do the same thing with a vaccination. And those data are used to judge the antigenic difference between strains. For example, there's a twofold difference or a fourfold difference between strains. And the bigger the difference, the more pressure there is then to update the vaccine with a newer strain, because the old strain is too different. So in our study, we've got human data on people being vaccinated for the first time, like ferrets would be in a ferret experiment. They're only given one infection. So we've got humans who are vaccinated for the first time in recent years anyway. And then we see their response against the vaccine strain, but also against the other strain. And so the left-hand side, two columns are H1 and the right-hand side are H3. And I think the numbers in this table, so this is, again, to be very clear, the first row is group 5 who got vaccinated in year 1. So the red is their response to the year 1 strain, the blue, so the off diagonal, the 40 and the 34.7 are the responses to the following year's strain, representing like a cross reaction to what's going to happen in the future, not so strong. And then for the bottom row is group 4 who got placebo and then they got vaccinated in year 2. This is their year 2 response. So it's the red is the homologous response and the black on the left is the cross reaction. I think there's something in these data to infer antigenic differences or distances between related strains that are included in the vaccines in drive only, not other strains that are not included. But that's as far as I've got in my thinking. And I'm wondering what to do. One of the wrinkles is that the largest number in this table is the bottom left off diagonal, the 180, that we can't really explain why there's such a strong cross reaction for the year 2 vaccinees who didn't get vaccinated before. They're not repeat vaccinees, but they had a really strong reaction to AOI. Maybe the antigen is more potent, and there's noise in this assay. So we will have more data in the future from a larger sample size. Maybe it's just an element of noise. But a lot of exciting things to learn, not only on the primary outcomes of the trial, but on other things we can do with the data. So I've finished there, ready for some questions, if there's any questions. And I would like to acknowledge support for the RETAIN study from US CDC, and then the DRIVE study from NIAID. Thank you very much.
So, this is a bit of a long-winded setup, so I apologize. But I did my PhD with Mark Lowe on adjuvanted influenza vaccine in children in Hutterite colonies in Canada. And so we found that like adjuvanted vaccination seems to overcome like vaccine mismatch. We didn't look at repeated vaccination, but I wondered like what your thoughts were, given that like T-cell correlates of like protection seem to be like implicated in older adults, like whether you thought that adjuvantation might overcome repeated vaccination.
Separately from the RETAIN and the DRIVE, so I have another trial in older adults in Hong Kong called PIVOT, which is a trial of alternative enhanced vaccines in older adults. So we've got 4 vaccines we're comparing: standard dose, adjuvant vaccine, MF59 adjuvant, not the one that you were testing, but another adjuvant vaccine from Sanofi. It was MF59, okay. Yeah. That was MF59, so same vaccine. The high dose vaccine from Sanofi, and then flu block like in DRIVE. And so that's a 4-year trial with combination strategies as well. And the headline findings, my interpretation so far, not yet published, is that the adjuvanted vaccine does very well because it gives a broader response, which covers mismatched strains. And this is a very interesting observation that the other vaccines are not so able to do, but the flu block does very well against matched H3 and 2, which is a big worry in older adults. And the high dose vaccine just generally gives a very good immune response. So I suspect when we come to write the paper with the results of that study, we may have something good about every vaccine. Every vaccine's got its advantage. There's no single winner. But I think the adjuvanted vaccine looks to have a number of advantages, and the cellular response as well is stronger and broader. One of the other things we did in the PIVOT study was have like a mix and match over a number of years, so have combination strategies. And one of the slightly worrying observations from the preliminary data we have so far is that people who got an enhanced vaccine in the first year had a weak response to the standard vaccine if they went back to standard the second year. The response was really attenuated. And so we see repeat vaccination effects for all the vaccines. And I published a paper recently about the enhanced vaccines where still have repeat vaccination effects, but not as strongly as standard dose. But then if an older person's been getting an enhanced vaccine, and then for some reason they go back to a standard dose vaccine, there's a really weak response after they've been having enhanced vaccines. So that's a concern that, if there's a policy decision to change to enhanced vaccines, like in the US and some other parts of the world, we have to be very careful about people not then going back, because it really won't work very well, at least for the first year. I would like to do more studies with the adjuvant vaccine as well. So, we'll see. But I think it's got a number of advantages. Yes, thank you. Yeah. Yep.
Can you comment about the effect of age on the antibody ceiling and the effect of the
Sure. So we have two different studies here. The retained study is a study in older adults, and there's often an assertion that older people have weak responses to vaccination because they've got immune senescence, they've got weak immune system in general. We don't really see evidence of that. Actually, the responses in older adults have been quite good the first year, but then in subsequent years, they have weak responses, but they're getting vaccinated every year. So the ceiling effect and the repeat vaccination effect combined seem to be particularly noticeable in the later years of the retained study. One of the things I would like to do is somehow put a side-by-side comparison. There's some overlap in terms of the years of these studies and the duration of these studies, and see whether the strength of ceiling effects or the strength of repeat vaccination effects is similar in these older adults who maybe have, to some extent, a weaker immune system, versus the younger adults in DRIVE where we're a little bit stronger. But that will have to be in the future. What I can say for now is that we see in both groups attenuated responses to repeat vaccination, and we see a clear evidence of ceiling effects as well.
So what you're saying is the two age groups from the two different studies get similar.
I couldn't tell you how similar, but they certainly both have these effects. Yeah, to what extent they're similar, I think remains to be seen. My suspicion is the older people will just be closer to the diagram in general, just having generally attenuated responses. For various reasons. It's not only their age, but also their immune history. They would have had at that point more infections, more vaccinations.
Thanks, Ben. Awesome, as usual. I had two questions that sprang to mind. The first was the comment about RT at the start of the season.
Yes.
Which you commented was about 1.3 or whatever, which actually we're just talking about. What do you think the potential impact is of having no exposure to influenza for the last 2 to 3 years on that relative to usual season?
Good question. So, I have another study, a cohort of about 2,000 people recruited in 2020 and followed throughout this period with blood draws every 6 months. And we have data in that cohort in people who didn't get any flu vaccination, no infections, about the antibody levels against vaccine strains, which were the strains that we were worried about for H1H3 and the 2Bs. There's very little waning across this period in antibody levels, which is surprising. So after vaccination, you get a big boost. I can show you an example. You get a big boost and then it comes down. In the people in the cohort that I'm describing, essentially when you get you come down after the vaccine being infected, it flattens out and it goes kind of horizontal for quite some time. It doesn't keep going down. And so in a way, what I think may have happened in the COVID pandemic is that the level of population immunity against the strains in 2020 stayed relatively the same, and the viruses in circulation in other parts of the world didn't evolve as much as they would ordinarily have done over a 3-year period. So it was On
Is actually no. I mean,
I think so. I mean, I don't know it myself, but I think so. I think people that study the virus evolution would be able to say whether there's been a similar speed. But my understanding is that it's been slowed down, like on YouTube when you play the video at half speed or whatever, it's slowed down. So the evolution has not been as fast. And so if we ordinarily think that new flu epidemics occur partly because of a loss of immunity and partly because of immune escape from the virus, I wonder whether we could learn from this experience that immune escape is a relatively more important phenomenon. And if the virus doesn't have much evolution, then in 2023, when it comes back, because it's quite similar to what it had been before the pandemic, the population immunity has not actually changed that much, hasn't been that much waning. So again, to emphasize, in the short term, after you get infected or vaccinated, you have a boost and a drop, but then that stabilizes. And in the longer term, after say 6 to 9 months, for the following years, doesn't change that much. And this is a unique period because in any other period of time when we have cohort data and cohort studies, there's flu and people get infected and that confuses the picture. But we have a situation where we have 3 years with no flu. So it's very exciting to have antibody waning data in the absence of any exposures and any vaccinations as well, of course, in that particular cohort. So that's going to come out soon, I hope.
That's fascinating, and it relates to the second thing I wanted to ask, which was later, which I was just going to ask about correlates of protection.
Yep.
Where you think they might be.
For flu.
Yeah, and relating it to what you've been talking.
Yeah, so one of the things we can do straight away is we know there's as I go back to the retain study, we know that there's a curve that relates the antibody level to the presumed level of protection. That's a curve that's used for vaccine licensing. There's quite a number of studies establishing that a titer of 40 is roughly equal to about 50% protection.
Against symptomatic infection.
Against symptomatic influenza, maybe infection, but against symptomatic influenza, a number of different studies in different age groups pointing in the same direction. And so if we were to use that curve and translate the red and the blue with that curve, that's where I'm getting my estimate that there's probably a 10% difference in clinical protection. But in reality, I think there's more going on in the immune system than just the antibody level against the vaccine strain. And in one recent paper, my colleagues and I estimated that the HI titer explains a half to 2/3 of the protection from vaccination. And there's other studies coming in, including one from Mark Loeb, actually. I think he's maybe you, but someone in Mark Loeb's team has done the same kind of analysis for one of their studies. Well, there you go. So there you go. So I have seen that somehow. I've seen it somehow. And again, there's a mixed picture. The HI titers do not explain fully protection. There's other things in the mix, but it's very difficult to identify what those other correlates are. And that's just talking about correlates of protection against infection or symptomatic infection. When we think about what protects against severe disease, that's very unclear. Very few studies on that. And it's an area I'm very interested to do more on. I have some grants on that exact topic, but it's difficult to find data that would inform it. One of my ideas has been to look in vaccine effectiveness studies. So in these type of studies, we enroll people at the beginning of the year. We get their blood samples, we see what happens to them, and we may pick up some infections, but we won't pick up severe cases. But another way to go is that in reverse, you find people who are symptomatic with flu or even hospitalized with flu, and you get their blood as quickly as possible, acute sera, where the antibodies hopefully haven't changed that much. Maybe other things you measure in the blood haven't changed that much. It's too early in the course of infection. And maybe looking at those type of samples in a case control approach.
Or test negative.
Yeah, test negative, exactly. So you'd be able to see some differences between other immune markers that might be reflective of protection against severe disease. That T cells is often presumed to be one of the correlates for protection against severe disease, but there's no studies to establish that as far as I know. So that's another interesting area of research where we're stuck at the minute with antibodies. And for COVID, I can share with you a completely separate thing that I don't know if Nancy Learn presented when she was here, but we have in Hong Kong mRNA vaccines and inactivated COVID vaccines. And Nancy and I published a paper in Lancet Microbe last year where we show that the neutralizing antibody is it doesn't get boosted by inactivated vaccines for COVID, but inactivated vaccines work very well for COVID, despite not stimulating a neutralizing antibody response. So whereas we think for COVID, the neutralizing antibody titer is the correlate protection for vaccines, that seems not to be the case, at least for inactivated COVID vaccines. So one more time, inactivated COVID vaccines work very well, but they do not stimulate a good neutralizing antibody response. And there's a number of different studies that demonstrate that, not just this one study in answer micro. So that's another area separate to this on COVID about what's the correlates of protection for COVID. Again, probably not just neutralizing antibodies. So a lot of things on the go at the same time to keep an eye on.
That's one question.
Go on.
So, I think the COVID lab, I released like paper a couple years ago about age, like birth year and H3N2 susceptibility and
Yeah.
the dynamics between your original exposure and then how you were protected later throughout life. And I wondered if there was a way you thought about integrating that with your like older adults in the case of H3N2.
Yeah, so it is in my mind. In the other cohort, we have a slightly wider age range, and there's a point of time, I think it's 1968 or 1957, where people were born before or after that. That's really the interesting timeline. So my eye is on that, and one of the things we're able to do is to do the antibody landscapes, where we look at not only the immune responses to the recent strains and the vaccine strain, but also the immune response to early strains. And I suspect that with the landscapes, we could pick up some information about early life infections. For example, you see someone getting a vaccine with an H3N2 strain now, and they have a response to a 1968 H3N2 virus, indicating they were probably infected with an early H3N2, or not if they don't. So that's in my mind to pursue in the future. Yeah, so far I have no such data on that type of thing. It's just something we plan to do in the future in our older adults. Yeah, it's a very interesting question. Yeah.